Fechamos bocadinho a porta. Good morning, everybody. Thank you so much for being here to the Faculty of Science, Department of Biology, but also in online, because we are now transmitting this seminar in biodiversity and evolution on the scope of the CBU, Biopolish Research Center, but also in the PhD program in biodiversity genetics and evolution, as well the master in biodiversity genetic evolution. We are organizing the seminars in biodiversity and evolution. And today is a special day because we have a special guest, a person that is really has done an amazing work. He's also I have a privilege that he is my friend, José António Godoy. José António Godoy is a senior researcher that one of the world famous biological stations that is in Doñana, in southern Spain, and is conducting a work that is also an amazing work because it's one of the more emblematic species in the world, that is Iberian lynx. You know that Iberian lynx also touched to us as a Portuguese because the Iberian lynx is Iberian species. Iberian lynx was extinct in Portugal, but has done a lot of people has done a great effort to save the Iberian lynx. And José António Godoy is one of those people, but I also should recall that there are people probably you don't know, mainly the people that are younger. Luís Palma, for instance, is a researcher biologist in Portugal that has started conducting a big campaign that salving lynx da Serra da Malcata. So save the lynx from the Malcata mountains. That was probably the last population that we had in Portugal. I remember this because I was young like you are now here attending this seminar, and I saw this movement of saving a species. And the person that like and wants to work on conservation biology is good to have this flex. And the Iberian lynx is not only an important species for the ecosystem, but is also what we call in conservation biology a flag species. And what we are bringing here today, and also that this change of bringing a renowned scientist to the Department of Biology, where we have the bachelorate in biology, is to try to motivate young students and young biologists to these problems in biology. But of course, our master students in biodiversity genetics and evolution and our PhD students. I believe that probably our PhD students that are abroad are attending this. I think we have already over 25% online. It's video streaming as well. But I really hope that you take this conference as something that is going to motivate you, put you thinking that what we need to do for our planet, how we can do it, and sometimes just using flagship species is important. José António Godoy has been working mostly in conservation genetics, is basically using genetics and now even go deep genetics with the genomes to help the conservation of the Iberian lynx. And what is going to bring us today is cutting edge information on how this information on genomes can in fact contribute for the conservation of a species. And by conserving these species, we are conserving habitats. And conserving habitats, conserving ecosystems, we are contributing for the better world. And this is basically what I hope we are, for sure we are going to hear today. And of course, I would like to give a big thanks to José António Godoy for accepting being here, also for the PhD of Carolina yesterday.
Okay, thanks. Thanks, Paulo. Thank you all for attending. I really appreciate being here with you and being participated in Carolina's thesis. That was exciting, and it's always a pleasure to visit CBO. I always have a friendly atmosphere, exciting discussions, scientific discussions, so I feel at home. I guess that's my way of saying you can invite me more often, and I'll be happy to come. Okay, thanks. What I'll be telling you about today is kind of a story, right? Like a movie. And like a good movie, it has a bad part where the tension builds, and then the outcome, the final part where the lovers meet, and everybody's happy. So this is something what happened with the Iberian lynx. And so my talk will be structured in two parts. One is more on how the tension builds and how the species approach extinction at the last part of the beginning of the 21st century. And then my second part will be more positive side on how the species is recovering. And my thread in this story is mostly based on the use of genetic information, more recently genomic information, in these two parts of the story, right? Okay. So as Paulo said, the species is now recovering, so that shouldn't be taken as a spoiler. After all, the story can be interesting if you know the end of the story. You went to see Titanic, right? And the ship was sinking. So this is kind of similar. You know how the story ends, but hopefully the story will be entertaining and instructive. Okay, and I also saw the casual here. I like that. So I guess it not only applies to dressing code, but to the style of the seminar. So being casual, I feel I'm happy if you interrupt at any time point and we convert the seminar in a discussion, right? My story is long, but I don't care if I think it's worth if we stop at any time you need some, or you want to provide some input or initiate discussion. So feel free to intervene. Okay, let's go. Okay, when we talk about genetics in conservation, there is this paradigm, okay, this concept, right? It's the extinction vortex. And let's go through this. So basically, genetics traditionally hasn't been part of the process leading species to extinction. No? We know the original causes, but that act on populations and species ranges to produce results in small populations. And these populations will eventually become small due to stochastic factors, etc. Right? So genetics is not part of that story with this scheme of things, but we know genetics plays a part when population becomes smaller. Okay, and that's because populations that become small, accumulate in breeding, loses genetic diversity. This results in lower fitness, so lower reproduction and survival, and reduced potential to adapt to environmental changes. So that maintains the populations in a critical or endangered status that will eventually result again in extinction. The one thing you have to take into account that this is a self-feeding mechanism. That means that even if the original causes of the decline disappear, once this mechanism is started, is functioning, then even if you repair, if you give habitat, if you eliminate the original causes of the decline, the species might still become extinct. So that means that you need to address specifically this process. And this, the motor, the engine fueling this, what we call extinction vortex, is made of evolutionary processes that gain relevance in small populations. And that's basically drift, right? Genetic drift. That's what's causing the diversity loss and the accumulation of inbreeding. So basically, with what you might know, diversity, reduce diversity, resulting in reduce adaptive potential, reduce heteroscedasticity, or increase homozygosity caused by inbreeding, reduce fitness, and that leads to extinction. Okay, we'll be talking about Iberian lynx. You may know, you may, this is a popular, I mean, well-known species. So some people seems it's receiving too much attention with respect to other endangered species. So I'm sure you know the story of the Iberian lynx. So I will not get much into detail, but I will focus on one aspect of the Iberian lynx that makes it particularly interesting for analyzing and studying the contribution of genetics to species extinction. And the point is that it's a good model to study these processes, because it went through a very steep decline, resulting in a critical situation. So it was really at the verge of extinction by year 2000. And one important point here is that the species is being managed quite intensively. And that's an opportunity in two senses. On one side, there's animals are being captured, managed, moved around, and we are receiving samples for genetic analysis. With some other species, sampling is difficult and is the main limitation. But more importantly, it's being managed, animals are being moved around, so we can contribute, we can make a difference if we advise those movement and that management based on genetic criteria. So we can really make a difference. Okay, and if, of course, charismatic, popular, flagship species, as you said, I'm not sure that's an advantage for the research on the species, but well, it has drawbacks and advantages, I guess. Good. Okay, well, yes, a reminder. So the critical situation was about the turn of the century, with only 2 populations remaining from a previously larger range in the past. And those 2 populations harboring less than 100 individuals. So really critical situation at the time point. Sorry. Usual suspects about the causes of this decline, habitat loss. Direct persecution has been important for the Iberian lynx, as for many other carnivores. And one particular aspect, characteristic of the Iberian lynx is dependency on rabbit populations. So it's a rabbit specialist. So if rabbit goes bad, rabbit populations will decline, Iberian lynx fall. And that has been one of the main causes of the recent, at least of the recent decline. Okay, one particular aspect where being a flagship species and iconic and charismatic species helped in research is because we got funded funds for sequencing the Iberian lynx genome. That came from private funding. And that was, in fact, one of the earliest genomes, reference genomes, to be produced for an endangered species. The genome was not that good according to today's standard, of course, so it was highly fragmented, but it was a good representation of the genome with good representation of genes as well. So why were we saying genomes were important? I mean, why we convinced people to pay to sequence the Iberian lynx genome? It's because we thought that genomics could really make a difference for species conservation. I'm not sure I will convince you about that. And there are still challenges and unfulfilled promises of genomics and conservation. We'll probably go back at the end, and maybe we can discuss that. But at the point at that time, we really believe it was a good idea, and that could contribute to species conservation. Okay, so first of all, as I told you, these processes result in loss of the genetic diversity and accumulation of inbreeding. So we had some data on that based on molecular markers that already suggested that the situation of the species, the genetic status of the species was bad. But for the first time with whole genomes, we had an accurate and unbiased estimate of genetic diversity in the species. And the surprise, well, kind of surprise was that the species harbored the lowest diversity ever reported for any species. Here is only a list of species that have been sequenced, whole genome. But if you expand to any, diversity estimates from sequences or markers or whatever, still is the lowest species. Well, there are a few more species that have also very low diversity. I'm not getting into what is the record, lowest diversity ever, but Iberian lynx is there. Remember that some of the examples of really low genetic diversity may refer to island populations, subspecies, but I'm talking about the whole species, okay? So that really makes a difference. And the other aspect that we were concerned with is inbreeding. And genomes provide a nice way of estimating, visualizing inbreeding. This is a chromosome, and you can plot genetic diversity along the chromosome, and you see these flat segments, right? Those are long runs of homozygosity that occurs just because an individual inherits the same haplotype from the father and the mother. So it's homozygote for a long stretch. So now this is an opportunity also to quantify, again, in an accurate and unbiased way, the inbreeding accumulated by individuals. And that was really striking to see that, especially the population of Doñana in the Southwest, one of the random populations, had a very high inbreeding coefficient. Some other estimates go up to more than 0.5. That's 0.25 is what you would expect if you made two full SIPs, right? So basically 0.5 is what you would expect if you are self-pollinators. So extreme levels of inbreeding there. Okay, and if you estimate the effective population size, that's parameter that allows us to predict what will happen in the future with the genetic diversity. It's very small. It's less than 10 for Doñana, less than 20 for the other random population in Andújar. That means that the low diversity that is present will be quickly lost in the future if nothing happens, if we don't do anything about it. So that's really worrying. Okay, but genome data also offer us the opportunity of asking the question, how did the species, the Iberian lynx, get to have this very low diversity? You can investigate that by inferring, reconstructing the demographic history of the species. So this is the more recent, this is the more oldest, and this is the effective population size. So well, no. So you see in red, more reddish color, the 10 Iberian lynxes we sequenced with the genome project, and this is a single Eurasian lynx. So you see parallel trajectories, but the Eurasian lynx has been have had higher population sizes than the Iberian lynx for a long time. Right? We applied other technologies that suggested that the Iberian lynx had gone through several, not only one bottleneck that we knew of in the end of the century, but older, earlier bottlenecks in the past. And in fact, those bottlenecks were not that population became small and expanded again, but the population became small, remained small until the next bottleneck almost drove it to extinction. And we can somehow date and size this bottleneck, the previous bottleneck, and it's occurred apparently around 300 or 400 years ago. Reduce the population, effective population size to 200 maybe. And then by the end of the century, those already, as I told you, that was reduced further to 1/10 of previous sizes. So we think that is this serial drastic bottlenecks that what resulted in a very low genetic diversity. Together with low population sizes for the whole story of the species, probably. So long-term population sizes. Okay, so in our next study, we wanted to kind of reconstruct this with more detail. So, and we had this opportunity by using museum specimens. So all the specimens, all the Iberian lynx specimens that we could characterize genetically and test the hypothesis is diversity was higher in the past or was already low by the time frame we covered. So museums provided samples, maximum 2 centuries. Mostly, I would say, most are samples from the few decades ago, from the 20th century. And we used different kind of materials, but we eventually got a good representation of samples across the distribution range in the 60s, more or less. So this is what's depicted here is what the range was in the 60s. So you see we have samples of all subpopulations and across a period of time. Of course, we wanted to go further back in time, and then we had to resort to these fossil or paleontological remains. Those are even more challenging than working with museum specimens is difficult, as you already know, Carolina already know, and Diana already know. But working with really ancient samples is even more challenging. So on two sides, it's challenging to get the samples and challenging to work with the samples. So yeah, we ended up with quite a few ancient samples of Iberian lynx across the Iberian Peninsula. Not all of them worked, almost none of them worked, but we were able to retrieve at least 10 mitochondrial genomes. It's not ideal. I mean, we wanted nuclear data, of course, but we couldn't get it at this point. Okay, and what we found is, what allowed us is to reconstruct the demographic and genetic history of the species in this time period, right? So basically, we came up with this hypothesis on this history of the species where, okay, now we have two different populations, highly structured, very genetically differentiated, and very small, okay? When we analyze this time period, the historical period, we see that there was already structure there. So many of these populations were genetically differentiated from each other. Genetic diversity was variable. I mean, there are subpopulations that had very low genetic diversity, even lower than the Ñana. There were subpopulations that had higher, in fact, the highest genetic diversity we ever found. And even those populations became extinct during the period. Okay, and what happened in the more remote past? So we had only mitochondrial DNA by then, but it was interesting to see that haplotypes, mitochondrial haplotypes were shared across large distances. So suggesting that probably the population was nearly panmictic in the past. So continuous range and dispersing individuals connecting different parts of the range. Okay, it's interesting to see that genetic diversity in the past was not that higher than in the present with mitochondrial DNA. In fact, historical diversity was higher than ancient diversity, but very little. So the difference was certainly higher than contemporary diversity. 3 haplotypes only in the whole species, no coding variation at all. So haplotypes are functionally equivalent. So, but okay, we were really frustrated to not having nuclear DNA from ancient samples. But we solved that more recently, okay? A few of those samples that failed, or we only got mitogenomes, we insisted, we worked on them, and 3 worked. 3 provided enough whole genome data. well, enough, some whole genome data. The amount is expressed in terms of coverage, how many times you read each base in the genome. So usually you want to read 20 or 30 times. Here we are left with 2-3 times, mostly, maybe 4. So there are low coverage genome sequences, but really valuable. Okay, they are from Catalonia, from Algarve, from here in Portugal, and from the Sierra Morena, close to Andújar, current Andújar. The first question is, how do those samples compare to contemporary samples? And it was nice to see that these were similar among themselves. I mean, they were not that different between them, and similar to those in Andújar, and very different from those in Doñana. With our reconstruction, we were hypothesizing, in fact, that Andújar, in fact, I didn't stress that, but you should have this in mind. Andújar is the remnant of the continuously declining population, right? It almost became extinct, but it didn't. And on the other hand, Doñana has always been, has been small for a long time. We dated that. So we dated about 200 years ago. Doñana was connected to this ancestral population, but has been 2 centuries being small and isolated. So that's what created the different genetic patterns in Andújar and Doñana. So under that hypothesis is reasonable that Andújar, the current Andújar variation is more similar to ancestral variation, right? And that's what we see here. Okay, so what would you expect after I told you this story? Ancient sample to have higher or lower genetic diversity? We were expecting higher diversity as well, but we were surprised to see that they didn't have higher, they even have lower genetic diversity. And we were surprised, and we made all kind of controls to see where are we estimating all right, the genetic diversity of these ancient samples. In fact, ancient sample provide errors, so you have sequencing errors that should contribute to increase. If you take that as real variation, you will increase the diversity artifactually. So it was already a surprise that being ancient, they had lower genetic diversity than contemporary. One other aspect is, okay, you have low coverage, so you are underestimating genetic diversity. So we reduce the coverage of the contemporary samples to make a fair comparison. And yeah, coverage, you see that coverage impacts genetic diversity. So the higher the coverage, the higher genetic diversity you estimate. But even if you correct for that, lower diversity in ancient samples still. Okay, that was a surprise. We couldn't explain that easily. So what kind of processes could increase genetic diversity from ancient to contemporary? Any idea? Mutation, no? But mutation is a slow process, and it's not likely to impact.
Probably, if you say the causal origins, but can you explain what probably we didn't catch? What you mean by ancient? Means only here. Probably it's important also for us to get an idea.
Yeah, that's a good point. It's not here. Okay, so samples were, I think this was the oldest, 4,000 years ago, 2,000, 2000.4. So few thousand years old. Yeah, I guess it's okay. It's written here. Good, but that's an important message. Okay, how long are we going back? A few millennia. Okay, so mutation not, but what about introgression? I mean, with which species? So which is the more likely source of introgression for Iberian lynx? Right, Eurasian is a sister species, right? Well, not really. Well, I'll try to convince you later that. But anyway, it's a neighboring species. And in fact, we know introgression has been an important process for Iberian lynx in the past. So I will show you later. So we checked whether contemporary lynxes were more admixed or less admixed than ancient lynxes. And concordant with the increase in genetic diversity, contemporary lynxes were more admixed with Eurasian lynx genes than ancient lynxes. That doesn't mean ancient lynxes were not introgressed. But they were less introgressed. So the thing is that probably in these 4,000 years, a hybridization event took place, an introgression event, and we still see those signatures in the contemporary genomes, and that allowed to increase the genetic diversity from ancient. It's true, we still don't have a direct link of introgression to diversity so that we can make a causal link, sorry. Yeah, sure. Okay, that's a good point, but that, yeah, in fact, as I said, that didn't, introgression, that introgression happened in the species didn't come as a surprise because there are ample evidence of hybridization between lynx and in fact between any felid species, and in fact any vertebrate. If you push a little bit further, yeah, we are finding introgression wherever we looked for, but felidae are particularly prone to introgression, to hybridizing between even distantly related species. And the genus lynx is one of the examples for this. Large amount of introgression. We already learned that with when we adjusted the demographic model to our early genome data. So we estimated both things were surprising. Early divergence, recent divergence, 300 years ago. Well, classical, we started assuming it has 1.6 or more or less million years ago of divergence with Eurasian lynx. But this suggests it was an early divergence, but more importantly, that the model was clearly supporting, so the data was clearly supporting a model with large amount of admixture in recent times. And in fact, we couldn't date this very precisely, but the data already suggested that the introgression happened even in the recent past. And we couldn't explore that it was occurring in the present. Of course, we know it's not occurring now, but probably to occur till very recent times. So I told you which is the sister species to the Iberian is the Iberian, right? Maybe not. So now that we have phylogenomics studies, so we can make phylogenetic trees with different windows along the genome or different genes. It's always the case that you get alternative topologies, right? And in the case of the genus lynx, the most abundant topology is in fact supporting Iberian lynx and Eurasian lynx as sister species. So the topology of this genus is this. So bobcat is like an early divergence, but then you have Canada lynx, Iberian lynx, and Eurasian lynx closely related, diverging around 1.6 or 2 million years ago. And the relationships are, well, close by, but if you say what's the most supported phylogeny in the genome is Eurasian and Iberian as sister. But there is a huge amount of the genome, a large proportion of the genome supporting this alternative topology, in which the Iberian lynx is older, an older species. So how it comes that it's now so similar globally? Again, because of introgression, a lot of introgression. So to the point that the most frequent phylogeny is the wrong one. It is misleading, the divergent history. How can we say that? I mean, how can we be sure if we have more or less the same, two alternative topologies supported by similar proportions of the genome? Because if we go to non-recombining regions of the genome, these are kind of free of admixture because selection will be clearly eliminate introgress haplotypes. So if you look at those regions, they will be supporting this topology. So it's not the most abundant, but it's qualitatively the more likely topology. So that means that the amount of admixture with duration link has to be huge. I mean, huge or extended in time since divergence. Okay, and you asked about this. How can it be possible? Because now they are non-overlapping species, but we know that in the recent past, maybe those 2,000 years ago, the species, as I said, was widespread in the Iberian Peninsula, but not only in the Iberian Peninsula, southern France, and even northern Italy, maybe. And the Eurasian lynx was widespread in Western Europe until a few centuries ago. And in fact, lynxes inhabited Eurasian lynxes inhabited the Iberian Peninsula until maybe a couple of centuries ago. We also sequenced an ancient Eurasian lynx genome in Iberia from the Iberian Peninsula. So yeah, there were opportunities. I mean, they live close together in the Iberian Peninsula, but as the range is expanding and contracted, probably there were multiple opportunities for contact and hybridization. Yeah, sure.
From the beginning, was like overlap to the speech in the extent which the expression is generalized towards the Iberian lynx. Did have there an effect of link selection, like that was like not only like impersonal introgression, but also bringing together other things that could also increase the diversity for the species nowadays. I don't know if that is.
I didn't get your question. So you're asking on selective selection on introgress?
Yeah, like the effect of link selection on the introgress region that could like not only keep the introgression in the population and persist in the entire species, but also bring with that other regions that could also increase the genetic diversity to the population nowadays.
Yeah, and so that the introgression will increase genetic diversity, that's kind of obvious because you're importing divergent haplotypes from different species. So in those regions that have been introgressed, the diversity will be higher. How long and what the selective effects on those introgressed regions after the introgression, that's, I mean, we haven't studied that, but yeah. So I don't know. Probably, I mean, the diversity should be increased in blocks, right? When you introgress haplotype enters the population, the diversity is increased for that region, right? But then recombination will eliminate probably what's deleterious, and the other will remain. And so what you are seeing is a process of recurring introgression, selection acting on introgression. So that's why we hypothesize that these low recombination regions are free of that mixture and then are reflecting a real topology. Yes.
And regarding those species, it spoke that the Eurasian lynx was in Iberia until a couple of 100 years ago. The range of the Eurasian lynx in Iberia over during this last stay until they were extinct in Iberia overlapped with the main populations of Iberian lynx, or was more near the Iberian mountains.
Yeah. Yeah, they were kind of segregated by habitat and climate. Makes sense. So the Eurasian lynx persisted in the northern mountains in Iberia, while the Eurasian lynx inhabited that. The Iberian lynx was more Mediterranean climate associated too. So, but you know, they have the immigration distance, which should be long enough to make them to contact eventually. So, yeah, in the Pyrenees, probably they also coexisted or co-occur for periods of time. So yeah, opportunities occur for hybridization. So, thank you. Any more questions? Okay, let's move on. Okay, so now we have this evidence that there is having loss of the genetic diversity, accumulation of inbreeding in recent times, and we have genomes. The genomes have been, as I showed, really useful to reconstruct the history, demographic and evolutionary history of the population. But this is, I was lying to you. I mean, this is a very simplistic view of what's driving the extinction vortex. Because we are ignoring selection, we are ignoring mutation, we are ignoring other processes, so that if we take that into account, the picture is a little more complicated than we were telling before. So basically, selection, well, has its own effect on genetic diversity, usually reducing genetic diversity. But the thing is that drift not only will lose genetic diversity directly, but also affect the efficiency of selection. So basically, a bottleneck should also affect genetic diversity in selected regions of the genome. So on the other hand, one of the results of this is that deleterious variation will not be efficiently selected. And that combines with an increase of homozygosity to result in this inbreeding depression. So why inbreeding is bad? Because if you have deleterious variants that are recessive and they become homozygote, then they are expressed, right? But another interesting process is that inbreeding depression is ultimately selection in action, right? So you are exposing deleterious allele to selection. If those individuals will have lower fitness, they will not transmit those deleterious alleles, and they eventually, theoretically, it's possible that the genetic load, that the amount of deleterious alleles becomes reduced after a bottleneck. Okay, so we have this more complex picture, and now we have the opportunity of addressing this with empirical data. But then we need population level data and analyze in a different way. So basically, we analyzed two studies. One was dedicated to explore how what we can call the landscape of genetic diversity. So if you plot genetic diversity across the genome, there will be peaks and lows, and selected regions will have lower diversity. So coding regions you're expected to have low diversity. Also, there will be differences among chromosomes, and along the chromosomes, different regions will have different diversity. So it's well understood, and it has been well described that this is a result of the combination interplay of all these processes, right? But often we think on equilibrium situations. So we have, that leads you to a picture of stable landscapes. But what we were wondering was how this landscape changes following a bottleneck. Because the interplay now of these processes is altered, as I told you. Okay, so we address this empirically by directly comparing diversity in Doñana, in a bottleneck, highly bottleneck population, Doñana, and I would say a non-bottleneck. You may argue that it's also a bottleneck, but as I told you, this is a very different demographic scenario, so we are considering this as a non-bottleneck. And to have replicates, we also use an Iberian lynx population having different demographic history. Again, 2 bottleneck population, 1 non-bottleneck population. And our approach was rather naive. If we calculate the diversity in the bottleneck, we calculate diversity in non-bottleneck, and we measure the difference, the relative difference, right? When we plot that in different features of the genome, regions of the genome, intergenic, promoters, or coding, or whatever. These are ranked here in order of increased selective pressure. That means that variation in these are not tolerated, so are selected against, usually. So that's, you expect low diversity there. And we plot this relative genetic loss when we compare the bottleneck and non-bottleneck. And there is an average. That's related, the average itself is related to the intensity of the bottleneck, right? That's reflected in average genomic diversity. So, for example, here, Poland and Kirov, I think it was 70-something% loss of overall genetic diversity. We have 80% loss. Oh, here it is. 80, 74, and 50% retention of genetic diversity. Okay. So we have an average. Different regions have different behavior following the bottleneck, but it was striking to see that the diversity was not lost in the high selected regions. How do you think that happens? That's especially, so you have CDS, for example, okay? Not so much diversity loss. So it's very more similar, the genetic diversity in the bottleneck to the non-bottleneck is ultra-conserved non-coding elements. Those are regions of the genome that do not vary across large scales, phylogenetic scales, right? They are highly conserved. So there, you don't expect a loss because there is no genetic diversity to start with. And what we see is that it's a gain in genetic diversity. Okay, so, and the amount of gain, that relative gain of genetic diversity is proportional to this, especially high in these ultra-conserved elements. So this is reflecting the process that we were discussing in the scheme, where drift is reducing the efficacy of natural selection and allowing deleterious alleles or moderately deleterious alleles that could be selected efficiently in large populations, in small population or bottleneck population, which have the chance of increasing in frequency even become fixed. So you see diversity where you should not see diversity, right? Okay, so that's a bad thing. So it's reflecting the accumulation of genetic load in small populations. Okay, but the other nice thing is that we can measure genetic load directly. So basically, for each segregating variant, we identify which is the mutation, the derived allele. So, and we count the number of alleles, derived alleles in different regions of the genome in different individuals, and we can average across individuals to get an average of the populations. And in accordance to theory, the number of derived alleles per individual genome should not vary in neutral regions. But what happened with the variation that affect fitness? So basically, we have a way of anticipating or predicting the effect of fitness of particular mutations with bioinformatics. Basically, well, it depends where the mutation occurs. Of course, if it's intergenic, we shouldn't affect fitness. If it's coding, they have higher chances. But even in coding, you remember, there are mutations that do not affect the amino acids, so it doesn't change the protein, so those should be neutral. But however, the changes in amino acids should have some weight, some deleteriousness, right? And bioinformatically, we can predict which changes will have a higher impact. So we can, non-synonymous variation, we can classify tolerated, so mildly, no effect in fitness, or deleterious. We predict higher than fit. In fact, we have other mutations that we know would result in loss of function. So by counting the number of mutations in different populations of lynxes, the same populations I told you about before, we see there is no difference in moderately deleterious, but with deleterious variation, there are lower amount of deleterious alleles in Iberian lynx and Eurasian lynx. That's purging, right? What I told you about. So it's an opportunity to select against deleterious alleles. So we have this double thing occurring in Iberian lynx and probably in other bottleneck populations. Some deleterious allele will increase in frequency, moderately deleterious allele will increase in frequency, and some high impact deleterious alleles will decrease in number because of purging. It's kind of contradictory, but not when you think on the dynamics of selection, right? And the other interesting thing is that we don't see any difference between bottleneck and non-bottleneck population. We see difference between species. So that means that the process driving purging is probably more related to long-term effective population size differences between these two species than to recent bottlenecks. Well, one clue that allows us to interpret this as selection, purging, is that it's not observed, it's only observed where selection is efficient. So centromere, low recombining region, selection is less efficient. You don't see purging there. Only, but in telomeres where recombination is abundant, the selection is efficient, you see purging. And you don't see purging in the X chromosome because why? Why do you don't see purging in X chromosome? Remember, we're talking about deleterious alleles that are recessive. That's one important message, because the probability of being purged not only depends on the impact, how deleterious the allele is, but also on the inheritance mode. So recessive alleles are purged, additive alleles are not purged differentially between large and small populations. So, but in the X chromosome, these alleles are anyway exposing in males, right? So they are no difference between large and small populations. Okay. So can we assume that populations that have been bottleneck are equally fit, the same fitness than populations that are not bottleneck? No, because the amount that the genetic load that is there is more expressed because of higher homozygosity in a smaller population. Okay, so basically you have the same amount of bad alleles, but higher chances that you express them. And that because of fixation or because inbreeding increase homozygosity. And that's where we now see differences with between different populations. Okay, oof, and it's almost 1 hour. Okay, so I'm reaching the end of my first part. Sorry, I was slow. But we can go fast through the second part. So the one thing that you may wonder from the start is, well, this is seeing, I mean, this is matter at all? All this genetic load or whatever, can you see any detrimental effects, anything in the lynxes that are not going right? Okay, and through the years, there have been a few things being observed and attributed to genetic effects, so inbreeding or whatever, without really evidence for that. But okay, since high prevalence of renal diseases, lymphoid depletion, cryptorchidism, high susceptibility to infectious diseases. In fact, this was showed quite drastically by single epidemics affecting Doñana a few years ago. A mild virus killed 6 or 7 individuals, and that was it. So seems like there is reduced immune response. And we have this epilepsy. It's epilepsy that occurs in young individuals, young Iberian lynx individuals. This has been observed in captivity, probably occurs in the wild as well, but it's difficult to observe because it's shaking, so the calves start trembling, shaking, and in the wild, probably they eventually die. They're treated and they survive in captivity. Okay, so that's circumstantial. I would say circumstantial evidence that something is not going right, especially in Doñana, because low reproductive success, large impact of epidemics. Here is a heterozygosity fitness correlation. That's the proper way of showing inbreeding depression is occurring. So you have lower fitness in more inbred individuals. That's a way of showing really inbreeding depression. We have little opportunities of doing that, but we could do that with semen quality, showing that in fact inbred individuals have worse quality than. Oh, sorry. Okay. I told you about epilepsy. The other things, we don't know nothing about the inheritance or if it's affected by genetics or not. With epilepsy, we did the right analysis, the proper analysis to show that it has a high heritability and it's inherited as a Mendelian, single locus Mendelian recessive allele. So which gene, we don't know. Okay, so basically what I told you so far is that lynx fits to this model of an extinction vortex. So what I wanted to tell you, and if you are willing to stay a little bit longer, I will tell you about the recovery part. Can we stay, Paulo? Okay, I will go quickly through this. This is a good part of the story, the positive part. But so basically, we have in this dynamics, and how do you get out of that? How do you improve the situation? So basically, with gene flow, okay, you want to add gene flow. If you add gene flow, inbreeding will be reduced, and diversity increased, fitness will be increased, etc. And so eventually the population will become viable. Okay, to summarize now this part, this is a scheme of what's the current conservation of the Iberian lynx. So this is Doñana population, the remnant highly eroded population. This is Andújar population, the second remnant, not so much eroded population. And all these things happened in the last decades with the Iberian lynx conservation. First thing that I must stress really hard, the first success in the conservation of Iberian lynx was that the population didn't become extinct. So remnant population survived. And that's only due to field work, managing habitat, managing prey availability, preventing persecution and non-natural mortality, etc. So that's what the first success of the Iberian lynx conservation story. Second part, a captive breeding program was created. One thing we needed to decide, well, it should incorporate founders from the two populations, or maybe only one, or keep them separated. But from the history I told you about, there is no reason to think that these two should be kept separated. Rather, as I told you, genetic rescue, I mean, getting out of the extinction vortex needs the two populations to be mixed. And that's what happened in captivity, okay? There are 4 breeding centers that are coordinated, working on a coordinated way, one of them in Portugal. So this is an international, all the Iberian lynx conservation has been international in the sense that it's Iberian from the start, basically. And this is genetically managed. And I will go back to that in a minute. The other thing that happened is that Doñana was highly eroded, and we had signs of inbreeding depression. So animals were moved from one population to the other. And this has increased diversity, has increased, in fact, population. So we think we are here an example of genetic rescue, a real genetic rescue. And we, in fact, found a link between increased population and improved genetic diversity, because we showed in captivity and in Doñana that F1 individuals, hybrid individuals, mixing the two populations, have higher reproductive success, okay, both in the wild and in captivity. And more recently, okay, population, this was not intended to be just a storage of animals, but rather a source for a factory of animals for reintroduction, if I can describe it that way. And that's happening since the 2015, more or less, where large efforts have been made to create new populations in areas where the species disappeared. Okay. And I'll jump all this to show you okay, to show you this. Okay, this is the current distribution of the species, 2022, right? So we had the remnant populations are still there, as I said, but they are larger, they are more abundant. These are populations that were created by reintroduction, but nearby the remnant Andújar population. Now they are working as a meta population. I mean, they are connected by gene flow, so we consider them to be a genetic unit for management purposes. But more importantly, there have been 3 new populations created, one in Algarve here, Valle do Guadiana. All of them are increasing now. All of them are, a lot of animals have been released, but the current growth is not dependent on releases. I mean, it's internal. The animals are surviving and reproducing actively, so numbers are increasing quickly. And in fact, animals have colonized the new areas. One of the questions was whether these populations will be connected or not. And we have so far anecdotal evidence that animals moving from one population to the other, establishing and reproducing. So those are good news. And in fact, the lynx project, that is the current project in conservation, was planning to create stepping stones, selecting areas optimal to connect two populations, releasing animals there. But in fact, they found that the lynxes will create this stepping stone by their own and select those areas. So okay, so basically this is happening. Genetics is helping with that. That's the part I couldn't tell you much about it. We are using genetic information to guide selection of founders in captivity, crossings in captivity, selection of animals to be released at each site depending on the genealogy and previous history, and we really think that this is helping because genetically improved animals with higher heterozygosity are behaving are having higher success and higher survival, so it should be good. Okay, and that's not the end of the story, of course. 1,600 individuals by now is good news. Not enough to say we have saved the lynx. Efforts need to remain till when we calculated what's the needed, how many lynxes were required for viability, not only demographic but also genetic viability, and we are in the terms of more than 1,000 breeding females. In the 20, we had already 300. Breeding females coming from the 2000 that were 20 or not much. So we have already increased a lot, but we still need to improve more. Okay, sorry for being a little bit late. You have questions and we have time.
Thank you so much, Jose, for this wonderful story that where we can use the genetics for preserving the species and lessons to learn not only for the past from some thousands of years ago to the present that we are serving. The time is already a little bit late, but I think there is room for one of the questions, not only from here, but also for those ones that are online. We still have over 20 persons online, so please, either here or please either, I don't know, and if you can add the chat or you can activate the micro if you want to put a question directly. But we want to start with the question.
It was a really great talk. It's good to see that the species is recovering and so well. And now that the species is recovering and we are protecting and including a large predator, it might have impacts again with people. So, and first, which is the plan of the project? To control these effects, which measures are you taking for avian invasion or other things are part of all of it. And second, how this might impact. So it will start to be the links of the fact thing, how it starts with the impact of the diversity and the species. Already several events, and it's not in the addition so it's starting to get better. It back.
Okay. Okay, I guess the two questions are first relating to the possible conflicts. It's true. Well, I must start saying that the attitude of people generally is very positive for lynxes. Part of the reintroduction program was to deal with that. I mean, to select areas where the attitude of local people was positive, and even educating and promoting a good attitude. So that's being managed, I think, quite efficiently. It's true that there are more lynxes now. Lynxes are moving. Lynxes are occupying unplanned areas and places, and eventually they are predating. They're predating on chickens, and they're predating in sheep as well. It's up to the administration to manage that, right? So it's through compensations usually. It depends, it varies between communities and probably between countries as well, how this is managed. In Andalusia, they're favoring, rather than paying for chickens, that could be, they are building enclosures that are safer for the chickens and avoiding predation. With sheep, some other communities are compensating damages. So yeah, I don't think this will translate in change of attitude or global attitudes. It will be local problems probably, and we are aware that persecution is still a problem. Illegal poaching is still a problem. We don't know how important that is because those mostly remain undetected in opposition to roadkills that we find them. So probably the impact of persecution has been, or poaching has been underestimated. Yeah. So, and the other thing is, well, how this will translate in viability in projections, and I don't see a danger from changes of attitude now in the global dynamics. Of course, genetics, your question about genetics, it will follow demography. So as long as the population remains large, the genetics will not be declining again. And the other part is connection. We need to be aware that for this population to be viable, they need to be connected by gene flow. So if that doesn't happen, then we need to manage. We need to move animals ourselves. And we're ready to do that, if we detect problems, local problems.
Okay, one last question. Anyone from online? Comment on the chat. Comment on the chat. You know, the person that made a comment on the chat, if you want to put the question directly. Was Gabriel, do you want to put the questions directly? Please, you can open your micro.
Do you hear me?
Gabriel?
Yes, do you hear me? Hello?
No.
No.
Okay, but I can read. So Gabriel say really interesting talk. Okay, I'm trying to speak, but it's not working. Okay.
Yeah, we can read it here. So.
Or you can read it. Yeah. Yeah, because it's good also to work to incorporate information regarding the purging of high impact deleterious mutations to the conservation and breeding program.
Okay.
Could the mixing of the different populations negatively affect the purging?
Okay, that's certainly a good question. So far, we estimated the quantity of genetic load globally and between populations. There has been this debate and whether when targeting genetic rescue sources should be selected specifically to avoid introducing new genetic load to a population that has been purged. So that's certainly a concern. I don't think that's a problem with the Iberian lynx because the two populations we are mixing now has a long shared history. So most of the genetic load will be shared. So probably no much new genetic load entering in the Doñana population if they or entering the captive breeding. How we manage that? So far, the population is being managed for overall genetic diversity, so increase genetic diversity and reduce inbreeding. So But what I didn't tell you about is that we need to go, well, it could be good to explore a step further in conservation genomics. I told you maybe the promises of genomics have not been fully fulfilled. I see the opportunity of genomics making a difference through the identification of local effects. So basically, what's the basis for inbreeding depression? What are the regions involved? What are the genes involved or the variants? Can we manage that to improve and avoid inbreeding depression? I think that's a qualitative step forward from the things we were doing with molecular markers. Because even though we are using genomics for management, we're using genomics so far the same way we were using markers, but with better estimates, of course. But we, in our project, new project, we are intending to, crossing that border will certainly require two things: extensive fitness information, individual based, and extensive genomic information to be able to link fitness and genomic regions together. And we are targeting that in the new project. So generating hundreds of whole genomes and using the fitness information coming out from the monitoring of the reintroduction and the captive and the remnant populations to get fitness parameters.
Jose, so I was planning to ask you that because I think it's not easy, not only question but also answer. So, but you are touching the point. So, do you think that if we, in the future, or the way just to make this risk of the genetic diversity of the Iberian lynx, that we should, for instance, take some genes from the Eurasian lynx and in some way try to incorporate on the Iberian lynx? This is in some way what you are saying that genomics can bring this opportunity to do. Because this is one of the big debates of genetic rescue, you know, and I fully remember when the decision of making a translocation between Andover individuals to Toniana, there was already a big debate, because I know that there was some morphology, the fur is different, more dots in some places, and people are saying, no, even the people in ecology, no, there are ecological adaptations, ecotypes, now you are putting everything together, we are mixing, so we are losing those archetypes. Okay, so now how far can we go into genetic rescue?
Yeah, well, that's a central, I could say, central debate in conservation genomics. So, yeah. So I'm saying that mixing is good, and I really believe it. But certainly, you need to say, what are you mixing, right? So. It should be different enough so that there is a net improvement. It shouldn't be as too much different so that you have a breeding depression. So mixing these two, even though there was some debate, but it was kind of obvious thing to do, the mixing the two remnant populations. We had this model of the Florida panther debate that was earlier than this Iberian lynx debate. So, but we knew that differences in pelage patterns were caused by drift. So the Doñana population had more variation in pelage pattern in the past, but it was lost. So it's often the case that people are interpreting, you know, two lineages, two populations to be genetically different, and you need to preserve them separately without realizing that they are different now because they became different because of the decline and the fragmentation and isolation. So, and not because they have been for long term that way. And you need to also select what you're looking for. I mean, traits that have no impact on fitness, you don't need to preserve those or base your management on those. And you want to fit populations. So I would encourage mixing, especially in this situation where differences are arising because of drift and isolation, and so we need to restore what was that. With Iberians, we were able to show that with the historical study. We clearly showed that it's 200 years that the two population has been isolated. So that's nothing in terms of evolution. We cannot think, in the past they were part of the single palmicted population, so there's no reason to think that there are adaptations or things to preserve. And the Eurasian lynx maybe too much. Right, okay, we also show that this thing happened in the past naturally. So okay, and the population, the Iberian lynx is still there, and maybe it's still there because it was introgressed in the past. So yeah, that was an interesting idea that the Iberian lynx survived just because it was rescued intermittently by Eurasian lynx. Who knows? But proposing that in that stage is not probably the time. I mean,
if we know, for instance, that you show a lot of the disorders, the diseases, and if we know this is that, then this is what genomics bring. Because if we know exactly what are the genes, the alleles that are provoking, for instance, epilepsy, and we can erase this from the breeding program, I think this is a major.
Yeah, I think that is a major thing. We're targeting that. We are really working on that now to try to identify these variants. There has been also some debate whether you need to manage genes. So this gene-targeted conservation approach probably is not a good idea generally, but if you identify this trait that is being genetically caused, that is being diminishing the fitness of the population, the viability of the population, and you can identify the variants associated. It doesn't have to be the causal variant, something that is linked to, you can manage to get rid of the disease without, importantly, without affecting overall genetic diversity and inbreeding. You can only do that if you have these tools, if you identify linked variants. In fact, the epilepsy is a good candidate. We already know it's inherited, and we already know the model is quite simple. Problems here, there are only 20 epileptic individuals ever, I mean, detected in the captivity. So that's quite a few to establish a association, a statistical association. But on the other hand, there are not more epileptic individuals because the disease has been already managed. Assuming it was a genetic disease, assuming it was recessive, and pairing of two carriers was avoided. So since we are doing that in captivity, no new epileptic individuals were born. So confirming that probably it's genetic. But at the same time, we are limiting the management options for genetic diversity. We are preventing mixing individuals that probably it would be a good idea to mix otherwise if you don't consider epilepsy. But with the variant, we could select even the offspring, the healthy offspring of a carrier, of two carriers, for example.
Thank you so much. This will be a lovely talk that touched half of conservation genetics area that I am my colleague
Yeah, I'm sorry, I didn't measure well.
So we have to invite
Yeah, that was my point.
We have to talk. So much for being here. We should not go further. We would like that we cannot. People are already writing yes. And thank you so much to those ones that are online. And for sure, we need to bring for them.
Yeah. Okay. Thanks.